an associate professor of biomedical engineering at University of Houston. So he received his PhD in biomedical engineering, Georgia Institute of Technology, and he did 2 postdoctoral researches. First one is Beth Israel Deaconess Medical Center, and Rice University, second one. And Dr. Kumar started his independent faculty career at the New Jersey Institute of Technology. Actually, he was my colleague during 2014 and 16 time at NJIT. His research focuses mainly on computational peptide material design, drug discovery, development, and delivery, biomaterials and tissue engineering, with a broad interest in information modulation and angiogenesis. He has authored over more than 60 peer-reviewed publications and hundreds of conference abstracts. He recently received an NHR1 grant. So there are many things actually, so we don't have time to explain all this. So warm welcome Dr. Kumar to deliver the talk. Thank you.
Okay, so thank you all very much for having me. Yeah, so come on in, get some food. Hopefully this is interesting. My name is Vivek Kumar, and as he just mentioned, I am an associate professor of biomedical engineering at the University of Houston. I moved there having spent 9 years at NJIT. So a little bit of background, because I didn't know. Just give everyone some background of where I came from. So I grew up in Singapore most of my life, which is outside the planet. And then I came here for undergrad at Northwestern, and I did research with this guy, Guillermo Amir. Since the time I did research on these polymeric scaffolds with him, he has since taken those polymeric scaffolds and taken them all the way to market. And Stryker, in fact, uses some of those polymeric materials for their implants. And just to give you an idea of how long this takes, that was 20 years ago. So biomedical innovation and development does take some time. Anyway, so I was there from 2003 to 6, and then I went to Georgia Tech to do my PhD. One piece of advice I give everyone, especially the undergrads who are doing research and the graduate students, I did small diameter blood vessel tissue engineering, right? Making synthetic blood vessels using biodegradable plastics at Northwestern. And then I did my PhD in vascular tissue engineering, which is making the same things out of bioinspired materials like collagen and elastin. What I would recommend to all of you is try to get a diversity of experiences. Don't do the same thing in both your undergrad and grad or even postdoctoral research. Try to get a breadth of expertise. Anyway, so towards the end of my PhD, Elliot, my PI, moved out to Beth Israel Deaconess Medical Center, which is one of the teaching hospitals at Harvard Medical School. So I finished up my PhD there, did a year more with him in Boston, and then in 12 to 16, I went to Rice University to work for a guy called Jeffrey Hartgren. I tried to branch out a little, so I did synthetic chemistry for microvascular regenerations, right? So small blood vessels instead of larger ones. Anyway, I think there's a lot of tremendous opportunities out there, so definitely get into the AI data science on whatever interests you and build up a variety of different skills is what I would recommend. Anyway, I was fortunate enough to get a faculty job at the New Jersey Institute of Technology, where 9 years ago I had a lot more hair, and then stress, 2 kids, life. So I spent 9 years there up until 2025, and I moved to the University of Houston. That was in September of last year, and since then, lost some hair still. So this is a stressful and demanding job. I strongly recommend getting on LinkedIn. It's a QR code to my LinkedIn profile. You guys should, you guys should definitely connect. One of the things that's really important in life, I realized, especially since after COVID, is the importance of social media, is the importance of connections, right? And I reached out to a number of collaborators during COVID, and you hear some of these stories in a minute. Is there water in the back by any chance? You just throw it up to me, or if not, that's fine. Okay, anyway, what was I lost my train of thought. What was I? Sorry? Okay, no, that's
hairline.
Yeah, we'll just go from the hairline. Anyway, so come back to my hairline. Lost a lot of hair, and hopefully this job gets less stressful. But do reach out, as I was saying about LinkedIn, making connections is really important. Ever since COVID, I made a lot of connections through LinkedIn, especially since we couldn't come to work. And we started doing computational peptide design. And that led us on a lot of different adventures into what we're doing today, which is novel drug design, as well as biomaterials design using computational methods. Anyway, so the problem that we're trying to address in our research at UH is the localization of signals. When you take a pill, when you inject a drug, IV, subcutaneous, what have you, it diffuses all around the body, and you have systemic side effects, especially if you're trying to treat a disease in the brain and the liver that's localized. What we want to do is to localize signals through with the use of materials. So essentially, there are a couple of different diseases, as was mentioned, that we try to target in the inflammatory or angiogenic space, right? Inflammation, you guys know inflammation. Angiogenesis is the lack of blood. Angiogenesis is the development of blood vessels. The disease this treats is ischemia, which is basically the lack of blood vessels. So as this video restarts, you'll see that basically there's a disease called peripheral artery disease, which is poor circulation in the legs. 1 in every 5 people above the age of 55, 60 has this, right? And that lack of circulation in the legs causes the calf muscle to die, and ultimately it leads to muscle atrophy and amputation, especially the toes and all the way up. So what we want to do is to develop materials that we can inject into the muscle that can locally stimulate angiogenesis to regenerate blood vessels where it's ischemic, where it's low blood flow. Now you can imagine that having that angiogenic drug, that pro-blood vessel growing signal all over the body might not be a good idea, especially if someone has cancer or something. You don't want those malignant cells traveling randomly through the body, or cancer cells. Anyway, another therapeutic that we developed or have tried to develop is an anti-angiogenic hydrogel. So this hydrogel kills blood vessels. Now one of the diseases, the disease that this aims to treat is a disease called wet age-related macular degeneration, or alternatively diabetic retinopathy. All those words means there's a lot of blood vessels that grows on the retina and you can't see anymore, right? Because of diabetes and things like that. My dad has this. And basically what he has to do is go to the ophthalmologist and get an injection into his eye every month, or every 3 months or so. So we tried to develop a hydrogel that can slowly release an anti-angiogenic drug, or itself is anti-angiogenic, and essentially reduce the frequency of injections to once every year or once every 2 years. Anyway, and other technologies. So the problem we're trying to address is localization of signals. So our solution, how we're addressing this, is using self-assembling peptides. So this material platform has different amino acids that has a hydrophilic, a hydrophobic phase, and as you just saw, a bioactive domain, these green domains on the edges. Now in aqueous solution, in water, they form these sandwiches like you just saw, and they form these multimers, a whole bunch of them stuck together, okay? Because of hydrophobic interactions in the gray region, right? A little bit of orgo, a little bit of chemistry here, yeah. The ends of these fibers have these bioactive domains, and we can do a variety of different chemistries to show that they form antiparallel beta sheets, both at low concentration and at higher concentrations, especially when we add ions like salts, like phosphate ions, like these little yellow balls here. They start to extend into long-range fibrils and fibers that entangle into hydrogels, okay? So basically we made a peptide that in water aggregates into fibers that makes a gel. And we can simulate this, we can do solid-state NMR to study the kinetics of this, as well as exactly how the fibrils form. And like I said, these single peptides that we design then aggregate into fibers that form hydrogels that we can characterize, and they look like this. They're nanofibrous hydrogels. So we can do scanning electron microscopy, right, using electrons to image scaffolds, fibers, and then we can look at the fibrous morphology and see that these are nanofibers that enhance, that allow cells to proliferate and grow on them. We can do atomic force microscopy to characterize the dimensions of these fibers, and they're approximately about 2 to 4 nanometers tall, 8 to 10 nanometers wide, ribbon-like fibers that entangle into gels that look like that, okay? Now, going back to chemistry, all those bonds that hold these fibers together are non-covalent, right? Van der Waals interactions, hydrophobic interactions, ionic interactions, hydrogen bonds, things like that, non-covalent bonds, which means these gels can break and reassemble very easily, right? Those non-covalent bonds can break and reform, right? Straighten your hair, right? Hydrogen bonds break, and then the next day it curls up again, or what have you, because those hydrogen bonds reform. Anyway, so all that means to say, we can syringe aspirate these gels and inject these gels, and they reform a gel where we put them. Yeah? Yeah, I'm feeling a little bit not. Okay. And we can test this with oscillatory rheometry, right? So at low shear, right? So tension, compression, rheology, right? Rheometric testing. Anyway, so twisted, shear testing. Anyway, so we can shear at low shear rates, and we can see that these gels are pretty gel-like, pretty solid. But at high shear rates, they shear thin, they liquefy, right? Not magic, it's like toothpaste. You squeeze it, it flows, but on your toothbrush, it stays there, right? Shear thinning fluids. Our gels are a lot weaker than toothpaste. They're more like Purell, or like off-brand, poor quality Purell, yeah, that kind of thing. They're weak, relatively weak hydrogels, in the hundreds of pascals range, to be specific. But basically, they shear thin and they shear recover. So you can pull them in the syringe, inject them, and they stay where you put them for about 3 to 4 weeks. And we've done this with mice, rats, dogs, pigs, and shown that we have a platform that is dentinogenic, helps dental pulp stem cells proliferate, that we can load cells into this and write different patterns or deliver cells into the heart, for example. We can make versions that kill blood vessels, that promote blood vessel growth, that bind viruses, that kill bacteria, and I'll tell you about some of those things. But the first thing that we do is that we design them, right? So we have the self-assembling domain that forms aggregates and that forms fibers. We've got time. That forms aggregates and that forms fibers. And we have that bioactive domain, right? That domain that activates the receptor, kills, promotes angiogenesis, anti-angiogenesis, or what have you. Excuse me. How we find those domains is that we look at known interactions of growth factors and receptors. So for example, there's a growth factor receptor called vascular endothelial growth factor receptor, which binds vascular endothelial growth factor. So VEGF binds this receptor, and you have a whole bunch of blood vessels that grow, okay? Easy. So we look at that interaction of how VEGF binds its receptor, which is a homodimer, and we look at the binding pocket, exactly where that interaction happens. We isolate the binding pocket and those specific amino acids, right, protein amino acids that make up that mimic, right, of the growth factor, and then we add those mimics onto our peptides. Right, that's what we did. So basically we Googled, right, a bunch of mimics of growth factors and stuck them onto these peptides, which is the reality of what we did. And nothing to do with ChatGPT, except since COVID, we asked the question, well, instead of just taking existing domains, like mimics of existing growth factors, why don't we use computational design to rationally design or better design these mimics so we can optimize and finer tune angiogenesis or whatever property we're trying to build. Right? So we've used a variety of different tools, some of which I'll tell you in a minute, to optimize the binding, optimize the sequence to promote binding into the receptors. What's also cool about this is that because these peptides, you guys with me so far, right? Peptide, put a domain that binds stuff, right? So the nice thing about this is that what you can then do is when you put these self-assembled peptides on top of a receptor, they assemble on top of the receptor. So when one of those mimics binds the receptor and it lets go, there's a mimic right next to it that can bind, right? So you have fibrillar self-assembly on top of the receptor, on top of the cells, on top of receptors, that promotes long-term durable signaling, right? So you don't just have signal on, off, you have signal on and it stays on for a little while. And we've shown this through a variety of different binding kinetics assays. There's one called surface plasmon resonance, where you take the receptor, put it down, you flow your stuff, it binds, you flow buffer, it releases, you get on-off kinetics. We flow our peptides, it binds, and it self-assembles, and you flow buffer, and it doesn't let go. So it's kind of like antibody binding, right? It's somewhat permanent binding because of binding and self-assembly, and when one mimic releases, the next one binds. And that's what we see through binding of our peptides to VEGF receptor 2, where you have binding, and when you flow buffer, it doesn't let go. And what we also see is super stoichiometric binding. So instead of one receptor, one peptide binding because of the mimic, you have one receptor, one fiber binding, right? So multiple peptides that then bind and let go, bind and let go. At least that's what we think is happening. And we've measured kinetics using both SPR and microscale thermophoresis, MST. So what we also think is happening and this is a little harder to do without like fancy imaging, is we think that these fibers, because they're long enough, these are nanometer fibers that assemble into sub-micron level fibers, right? That we can image through AFM and SEM. We think that basically you've got a cell, and you've got this fiber sitting on top of the cell, and it might be able to bind a couple of these receptors, bringing them together, right? To help with those, to help with the homodimerization to activate signaling. We don't know, we're trying to do some simulations, as you can see in the top row, to establish, or at least support that hypothesis. Okay, cool. So this is the super stoichiometric binding I was telling you about, right? So we showed with this drug called PCSK9. PCSK9 is an enzyme that regulates LDL receptor for cholesterol homeostasis, metabolism. Right? However, so basically there's a drug called Repatha, right, which binds PCSK9 and it drops your cholesterol. Better than statins, all that stuff your both parents are taking, right, every day. But it drops your cholesterol down to almost single-digit milligram per deciliter, which means there was concern that it decreases the fluidity of the plasma membrane, because cholesterol is important for plasma membrane fluidity and signaling. Anyway, it's a really, really good drug, so if you can inhibit PCSK9, blockbuster drug, multi-billion-dollar drug, this is like 2015 Ozempic, right? Not GenScript, Genentech made a peptide, or isolated peptide called PEP28 that they saw binds PCSK9 but releases, right? And they didn't have good kinetics, didn't have good stability in the body, in vivo. So we said, why don't we take that peptide, add it to our self-assembling peptide, and this was first when we showed that we can bind PCSK9, self-assemble on top of the molecule, and when you flow buffer, it doesn't release, right? So we have the super stoichiometric binding. And in addition to that, we have good stability in vivo. Anyway, there are many reasons, as we know, projects don't move forward, especially money and funding, so this is one of those. Anyway, so we've used this idea of the message in the medium, right? So the biomaterial itself has a bioactive domain that we can customize, that we can build basically how we want with today's tools. And within that biomaterial is the signaling domain. And we inject it, and we have that localized response. We've used this to build peptides. These videos should work, maybe. We'll build peptides that can bind SARS-CoV-2, as well as a number of peptides that can promote angiogenesis, blood vessel development. Okay, cool. We're doing great. Okay, so I'm going to take you through some of these technologies that we developed. So I'm going to start out with the angiogenesis story. So as I've mentioned, and hopefully this is now canon, we have the self-assembling domain that forms a fiber. We have these bioactive mimics, these shorter pieces next to it, that present the bioactive sequence along the edges of the fiber, right? So this fiber that forms the hydrogel has this bioactive epitope at very high density locally, right? So within this hydrogel, you got a bunch of fibers that have this bioactive domain. We can make them fluorescent, we can inject them under the skin, and what's cool, I don't have this animated, let me just draw. Okay, what's cool is that this was a hydrogel that we injected under the skin in a mouse, all right? After 7 days, we took the hydrogel out, we flipped the skin, we took the bolus, the gel itself with the surrounding tissue. That's a glove on my finger with the hydrogel. And basically the surrounding tissue, and we did histology. And we stained for different cell types, looked at what's going on inside. So this area that I encircled in red was hydrogel that was acellular, that within 7 days had a bunch of blood vessels growing in. See this guy? All these red circles are alpha smooth muscle actin line blood vessels, right? So they're mature, small microvasculature. They're not just capillaries, these are 15 to 50 micron sized microvessels, arterioles, venules, that are stabilized not just by endothelial cells in green and platelets, which show up in the middle, but also smooth muscle cells. Anyway, so this was very exciting. We can cause localized angiogenesis, right? I showed this to a pathologist and they're like, is that cancer? I'm like, no, that's my hydrogel. It's not good. Anyway, so we thought this was really exciting because not only did we see a bunch of blood vessels grow in, we saw a bunch of other cells, right? These blue dots are nuclei of other cells that was associated with other tissue development, collagen tissue development as well. Anyway, so, oops, that's still there. How do I? Hold on, I can get rid of that. Okay. Okay, there we go. Start it again. Okay. And what's interesting is that depending on the type of hydrogel we put in, right? Whether it's a angiogenic signal or no signal or something else, we have different biological responses. And because these gels are injectable and retrievable, they degrade over about 3 to 4 weeks. We can retrieve them, take a look at the types of cells that go in, and depending on, as I just said, depending on the type of cells, we have different levels of CD31 positive endothelial cells that infiltrate, for example, and in data I was reviewing yesterday with my student, different kinds of mesenchymal stem cells, different immune cells that infiltrate over time, right? So we can chronicle this and try to tell a story, right, which is what science is about, and life, right? It's about telling stories, of how the cells infiltrate these different scaffolds, or how different cells infiltrate these scaffolds. You got to reinforce the message, right? Just have it on the same slide again and again. Okay. Anyway, so it's pretty cool, right? So we made this platform that's pro-angiogenic as well as a variety of other bioactive domains that we've attached. And then we said, what different disease models can we treat? So for example, we treated diabetic infected wound ulcers. So when folks have diabetes, they get these wounds that heal poorly because they have angiopathy, neuropathies. They can't feel it well. The blood vessels don't grow well. They're old. I have challenges healing my own wounds. I get hurt so often nowadays. It's only pickleball. No more tennis. It's pickleball. Low impact. Okay, and then 2 days rest. All right, so we create these wounds, and then sometimes we infect them depending on the model, and then we inject our hydrogels and take a look at how quickly these wounds heal. And we've shown that these angiogenic hydrogels promote early angiogenesis, which decreases later scarring in the tissue, regenerated skin. We've treated traumatic brain injury. We've done a fluid percussion injury model with James Harwa, who used to be at NJIT a couple of years ago, and then we injected our hydrogels on top of the cranium, closed, and saw histologically, we didn't do behavior, and saw histologically at least that we had neuroprotection and decrease of secondary glutamate injury by chelating some of the glutamate with our hydrogels. Anyway, what's also interesting is that we took these hydrogels, shoved them into other materials, like we took porous plastics, the stuff I worked on in undergrad, right? It's only porous plastic materials. We took those, centrifuged in our hydrogels, and then implanted those implants under the skin to see whether we can improve tissue integration, right? Like any kind of implant you put in the body, your body is going to form a fibrous capsule around it. It's going to say, hey, that's foreign material, and trying to mount some kind of response, and ultimately scarring, if not more, against the tissue. So what we said is, if you can promote some kind of tissue integration to a porous scaffold, you can improve osteointegration, for example, or implant integration. So we took these polymeric scaffolds, put it in our hydrogel, and I don't have other slides of this, but if you look really closely, or just believe me, you can see in the middle of the pores, in the middle of the scaffold, millimeters into it, right? We have tissue growing in from the surrounding. And this is under the skin, and this tissue growing into these scaffolds. Anyway, what pays the bills? What paid the bill for a year, and it was hard to get that money, was a study where we injected hydrogel loaded with induced pluripotent stem cells, cardiomyocytes from induced pluripotent stem cells, into mice with a myocardial infarction, right? So tie up one of the blood vessels, causes the left ventricle to infarct, muscle dies, inject hydrogel, plus minus cells, and then show that we had better survival. What we're trying to pay the bills with, what we're trying to get some funding for is a peripheral artery disease model, where I'll show you some data where we made a couple of different hydrogels that preserve limb architecture, preserve limb, and what currently pays the bills, it's all about money, is dental pulp regeneration, where we take these hydrogels that are both angiogenic and antimicrobial, inject them into the teeth to help regenerate dental pulp. Specific to dental pulp, because this is a lot of what we do, in a pig model, we expose the dental pulp, we drill a little hole on the tooth, and after like 2 weeks, after 2 days, it gets an infection, causes inflammation. However, the nerve kind of dies, which is what happens to all of us, and then we go to the dentist and we say, hey, I think I have a problem, and they tell you through an x-ray you need a root canal. So we do an x-ray on the pig, see that it needs a root canal, and then 6 weeks later, extirpate, remove the dental pulp, inject our hydrogel, and then cap the tooth, just like you would do in a human being. What we then monitor, again through x-ray, is whether the lesion is healed, whether we treated the infection, and whether the pulp is regenerating. And then we take the tooth out, and then we do histology. Now we've done this in dogs which didn't have the infection. We just took out the pulp hydrogel cap, right, a couple of years ago. And what we showed in dogs is that you can regenerate dental pulp, which is the soft tissue inside. The hard tissue is the enamel and dentin, right? We can regenerate soft tissue all the way from the apex, which is in your jaw, to the crown, the top of your tooth, right? And we can regenerate tissue that not only has blood vessels, so you can see this nice oblique section of a blood vessel with a lot of red blood cells, but also nerve bundles and the cells that touch the inside dentin, stain positive in dental protein, and have these tubules that go into the dentinal surface, the dentinal tubules that go into the dentin surface that anchor the tissue and support that, tooth growth, essentially. So we can regenerate dental pulp, and we're trying to do an infection model, right? Take home message. So what's also really exciting is some of the work that we're doing in a hind limb ischemia model. So here we tie off the blood vessels to the leg, one of the blood vessels in the ischemic, low blood flow ischemia side, this side. And right after we tie it, you can see there's very low blood flow and laser Doppler perfusion imaging, right? Like ultrasound, with color. So you've got this blue low perfusion region, and what happens when we inject this low perfusion region with saline or just the mimic domain, right? Not the self-assembling peptide, just the mimic that diffuses away very quickly, or whole growth factor that diffuses away very quickly. What we see is that the limb starts to fall off, right? So fewer toes, no limb, right? Again here, loss of digits, loss of toes, loss of muscle tissue and architecture, as you can see here, and saline, that's just terrible. However, when we inject our self-assembling peptides, we see that we have very good preservation of limb architecture. We put these mice on treadmills, and we show that they run just as far as they did prior to surgery. Okay, so some work that we published. So you're getting this idea, right? You have a self-assembling domain, you had a bioactive mimic, and that promotes signaling locally. So come COVID, so this is probably a little bit earlier in the overall story, but come COVID, we asked the question, how do we get back to work? How do we get students in the lab so that we can apply for some grants? Because we have to shut down. And if you're working on a COVID project, you can come back in, right? So we said, we do COVID research. So the structure of the spike protein, which is the receptor binding domain specifically of the spike protein of coronavirus, was elucidated, right? The part that binds human ACE2, angiotensin converting enzyme, 2 receptor. So basically, what we did was we designed a peptide that binds the spike, that binds RBD, that binds spike, and we attached a self-assembling domain. So the idea would be that it binds and creates this fur ball, this fuzz on top of the surface of the virus, preventing the virus from attaching, preventing the viron from attaching to ACE2, right? So that was our hypothesis, and we developed a peptide that did that. First by computationally modeling self-assembling domains, and then modeling self-assembling domains along with the binding sequence, and then doing some molecular dynamic simulations. Now, we're no expert in doing everything. We work highly collaboratively with a lot of different experts around the country. So this specific collaboration on the MD simulations was with Peter Krau, for example, at UIC. Anyway, so we synthesized these peptides, and we showed that these peptides can bind not just pseudovirus, but also live virus, and significantly reduce viral burden. Okay, hold on. Viral titers. Okay, so that was the end of that story. So yeah, significantly reduce viral load. However, we did that in vitro. The idea in vivo was to make a molecular mask, right? So make a gel. We make gels, right? Make a gel that you can spray into your nose. So it creates this gel layer that prevents the virus from hitting your nasal epithelium, which is where a lot of infection originates. We tried doing that in mice. We tried doing that in a golden Styrian hamster model, which is the primary model for COVID. Except, as all of us know, research takes time. And as an academic researcher, we started doing those in vivo studies in mid-22 or late 22, something like that, or maybe early, something like that. And the type of funding for COVID research rapidly dried up once vaccines came out, and all of us, no longer had a big COVID problem, and it wasn't so much of and the funding moved to more long COVID. So anyway, long story short, we published a nice paper, but we didn't move forward with creating pan antiviral. therapeutics that we proposed. However, what we also work on is not just killing viruses, killing bacteria as well. We have time, right? Yeah. So you guys know canned food, and canned food stays good on the shelves because it has a antimicrobial, and sometimes it has an antimicrobial called polylysine, right? A whole bunch of positively charged lysine, so positively charged amino acid. What that does is that it interferes with bacterial membranes and pierces them, right? Because bacterial membranes and biofilms are highly negatively charged. So it goes there and pierces them and causes them to lyse. So it's a good antimicrobial, it's a good preservative. Not great for the human being, not great for biological, biomedical applications. So what we said was, can we use this idea of polylysine, because our peptides have lysines on the ends, right? Lysine, serine, leucine, a bunch of times in lysine. We said, can we increase the number of lysines on the edges, on the ends of these peptides so the blue here is lysine, that's not playing, and the green here is the serine, leucine self-assembling domain. I tell people, if you get 30%, that's good. Oh, the movie doesn't play, that's okay. But basically, we did an MD simulation, a molecular dynamic simulation of a sandwich, a bunch of these peptides with lysines on the ends, 6 lysines on the ends, and self-assembling domain in the middle, on top of a bacterial and fungal membranes. And we showed that these peptide fibers bind to the membranes as well as integrate within the plasma membrane and start forming pores. I should update the slide. We have better simulations that show that they start forming pores, carrying water into the bacteria, lysing them. What's interesting is that this is a physical mechanism of action, right? So this is not some drug that hits some enzyme or some gene target or something like that, which the bacteria and virus can evolve around. This is their membrane. It's very hard to evolve around not having a membrane, right, for bacteria and viruses. So what's interesting is that we've tested these hydrogels that are antimicrobial against a range of bacteria and fungi, both resistant species and regular species, and in multiple passages, right, to show that they remain efficacious both in planktonic as well as in biofilm form, right? So even when they are individual cells or they're covered with this calyx that protects them from conventional current antimicrobials and antifungals. So we're really excited about that. We're trying to use these antimicrobial hydrogels as well as angiogenic hydrogels, like I mentioned, for dental pulp regeneration, for a variety of surgical applications, as well as wound healing applications. I'll be done soon, right? So if you're bored, we'll end soon. All right. So one of the really interesting models that's going on right now, we've got some really cool data that we're trying to get a larger sample size, larger N for. It's a cecum ligation puncture model. So in mice, they have a cecum okay, let me back up. So the disease we're trying to treat is, or the problem we're trying to address is secondary surgical site infections, right? So almost any surgery that you have, be it abdominal, aortic, hernia mesh, which is the most common general surgery procedure, dental, whatever, right? Let's say abdominal procedure. Any abdominal procedure you have has a 7 to 30% chance of infection risk from bacteria on the skin, from the surgical implant, from a variety of different sources. So we asked the question, can we use these peptide hydrogels, which are sprayable, which are biocompatible, which kill bacteria, can we use it in a surgical model where you basically, right before you close, you spray this hydrogel and then close? And we asked ourselves, what is the most severe sepsis model, right? What is the most severe infection model that can simulate this in the worst case scenario? For example, and this is a true story, one of the people who was in my group, because we moved, not everyone moved. One of the people who was in my group, his partner had had a bowel obstruction and had to go for surgery. And basically, they separate the bowel, remove obstruction, put it back together, right? But that put it back together needs to be perfect, because if you have any holes, any I don't know, gaps, what have you, in that anastomosis, that connection of the intestine, it can cause leaks, that can cause infection, that can cause sepsis. And lo and behold, his partner had sepsis and had to go back to the hospital like 6 times to figure all that out. So we asked the question, can we make a spray that you just spray into the abdominal cavity and close so you can prevent secondary surgical infections? Now keep in mind that almost always, if anyone's had surgery, you prophylactically take antibiotics. You're like, just in case, let's take some antibiotics. So this can reduce that antibiotic or augment its effect. Anyway, So the model we chose to use was the sepsis model, which is a very severe model. Mice have the cecum, which is like the end of the intestine. There's a little extra part that helps them digest fibers, I guess, because they eat a lot of fiber. So we take a needle, pierce the cecum, ligate the cecum, about 1 centimeter from the end, pierce it about 5 millimeters from the ligature, and then spray our hydrogel, because these are sprayable gels, right? Shear thinning sprayable gels, and then put it back in. We stain the gels with 0.01% methylene blue, which surgeons use for visualization anyway. So we stain it with that, which is why it's kind of blue here. And then what we see, at least in 1 week studies, is that these hydrogels have at least 1 week survival when untreated animals or control treated animals typically have sepsis and die within 1 to 3 days. So this is the idea. So I put this idea into Google Neo, what's it called? The one that makes videos, right? So I made a little video of procedure. I'll show it again. There's so many weird inaccuracies in this, but I thought it was a good representation. Spray a gel and before you close, right? Anyway, that was the idea. AI generated video, that one. But, and this is the crowd to tell it to, taking a product to market is not easy. There are many challenges, one of which is regulatory burden, regulatory strategy, right? And has anybody heard of NSF ICOR? You guys go Google NSF ICORPS if the program still is going. There are a lot of lessons that you can learn from the lean startup methodology and ultimately customer discovery. Basically, we took part in a couple of different entrepreneurship programs to help develop some of the products that we're developing towards the clinic, right? Because there's a different mindset you need. You know, as a researcher, you think about publishing a nice paper, but as a startup, as an entrepreneur, which I've done a couple of times and failed, so I teach about it now, several times. You have to think of a different mindset. What do investors want to see? They don't care about safety data. No, I take that back. They care about safety data, right? But applying to NIH to get funding, they're not super interested in safety data. That's not innovative and groundbreaking enough, right? So finding the right investment at the right time is important. And there are many components, one of which is regulatory strategy. And you learn this through customer discovery, going and speaking to these different people in the ecosystem of what you're trying to develop, right? So think about the end case, think about who's going to use it, and work backwards to you, and speak to those people, which we've done a couple of times. And what we realized is that before we go to the FDA and tell them, hey, we have this amazing drug that can kill bacteria and fungi, it's a sprayable, blah, blah, blah. The drug regulatory pathway is several hundreds of millions to bring a drug from ideation through clinical development all the way to product. And because of that, everyone tries to figure out a way to make medical devices, right? Because especially if you can find a predicate medical device, a product which looks pretty much like what you have for what you want to do, you can bring that to market very quickly and then do human trials and expand your indication, right? Say, oh, we can also treat this, right? So anyway, long story short, instead of pursuing a drug pathway, we've spoken to the FDA about trying to classify this material, scaffold, that has lysines on the end, that assembles the hydrogel that prevents infection risk, that also prevents infection risk. And we modeled this by existing 510Ks for a peptide called Pure Matrix. You know, some of you in biomaterials may have used that for cell culture. It's a similar self-assembling beta sheet peptide. Anyway, yeah, all these dates are wrong. Every time you put this up, you just gotta delay it because you never get those tens of millions in funding. You hope you will. Anyway, so as I mentioned, we've more recently, past 4 or 5 years, started to get into computational peptide design, as well as how we can use this not just for angiogenesis and inflammation and types of things that we've been doing for binding virons and viral particles and things like that, but can we also target different receptors? Can we target G protein coupled receptors, which I'll briefly tell you about in like a slide or two? Can we target sigma-2? Can we target other things? And to do this, we built an expertise in, first of all, understanding protein structures and their interactions, then using tools like David Baker at University of Washington and Dennis Hassabis at Google DeepMind, right? They won the Nobel Prize last year for AlphaFold from Google and Rosetta Fold. You guys should check out. There's a new Netflix show. It's called Thinking Game, I think. Right? Anybody know? Yeah. No. All right. Look it up. It's good. It's about this guy, the guy who made AlphaFold, and how they were competing to win this competition. It's really good. It's on Netflix. You should check it out. Thinking game. Anyway, they won the Nobel Prize because they figured out how to take an amino acid sequence of a protein, put it into a computer, and then generate a structure that's pretty darn accurate, right? Like 95% accurate to an actual structure. The actual structures you get through crystallography, through cryo-EM, provided different techniques, and there are databases of that. So they use machine learning to say, hey, look, this sequence generally corresponds to this kind of shape, this sequence to this shape, and they built that off of image generation, image recognition algorithms, right? I have a student that does this work, and he explained it to me like this, so I'm just conveying that part. All right. So we use these different modeling softwares to model the receptor if we don't know the exact structure of the receptor, especially if there are undefined parts. We then use docking software to design both linear cyclic peptides as well as small molecule drugs that bind these receptors and then generate novel candidates. Now you can imagine, I try to be specific here, it's very hard to do that AlphaFold, RosettaFold stuff for short peptides because peptides are floppy, right? When you have a long protein, as you guys remember, they adopt a secondary structure and then a tertiary structure, and sometimes partner, and a tertiary structure that locks them into a somewhat fixed position, right? A three-dimensional structure. And that you can model pretty well. But when you have a short protein, that can get floppy and doesn't have very good structure. So it's very hard to model that. Or there are many different conformations. However, what we can do instead is to make a cyclic peptide. Now a cyclic peptide, especially if you don't use glycine, makes it, if you use all D amino acids, for example, you get a chiral, relatively rigid molecule, right? Which you can then model better with. So anyway, long story short, we develop linear cyclic peptides as well as do small molecule screening. And it's not just, and we don't think it's sufficient just to look at docking or binding interactions. We also do MD simulations to look at residence time, right? How long do these molecules stick around in that pocket? And can we optimize that, especially since we look at self-assembly? Now all of this is possible because of gamers who helped develop the industry, because of the graphics processing power that we have today, right? So 5-10 years ago, doing 1 nanosecond, doing 5 nano, right? Second simulations, doing 20 nanosecond simulations was sufficient to publish well. However, when we submitted that paper to Nature Communications a couple years ago, they told us you gotta do at least 200 nanosecond simulations. And nowadays, when we apply for grants, we're saying we're doing at least 2 microsecond simulations. Now, all of this may seem like insignificantly small amounts of time, because doing more than that is very computationally expensive. But the rate that we're developing these graphics cards, right? These 4090, 5090 NVIDIA graphics cards, yeah, we've got 2 of each of them in the lab doing these simulations. The rate at which we're, basically developing computational power, we can do longer and longer and better simulations, more representative of native environments, right? Because instead of just looking at cartoon versus cartoon, we now can simulate solvents better, temperature better, membrane kinetics, a variety of different things. And we published a bunch of different papers on this as well. So we're trying to get, we just finished up this R63 one, this collaborative grant where we tried to develop novel peptide-based analogs to bind the sigma-2 receptor for opioid treatment. We've developed peptides that can bind based on consensus sequences of MS, multiple sclerosis antibodies, that can bind MS antibodies for treatment of MS. We've made versions that bind complement 5A receptor, MCP1 to modulate inflammation. And what I'm really excited about right now is where we're trying to modulate GPCR receptors by creating different kinds of decoys. We just filed, and we obviously file intellectual property on all of these different things. So sometimes we can't get into the finer details. Anyway, we're interested in making novel materials as well. As I mentioned, it is challenging. Oh, I don't have the video. It is challenging to simulate peptides because they're very floppy, but we do it anyway. So we're trying to generate novel materials that form different kinds of secondary structures, different kinds of architectures, and yeah. Anyway, and we've got a couple of different collaborations that look at other small molecules and peptides. And now we're biomedical engineers, so one of the capstone projects that I'm very excited and happy to talk about because we got a European patent on this as well, is a novel toothbrush that we came up with, okay? So this dentist comes, we do dental work. I presented at dental school. This is at Rutgers when I was in Jersey. And this dentist comes up to me, she's like, look, when I volunteer at patient care homes as well as pediatric centers and brush people's teeth with like either brushes or they put their finger with like this thing that brushes, they bite me a lot. And the other challenge is that they generate these aspirates that they breathe in and it causes aspiration pneumonia, it causes infection in the lungs. So is there any way you can figure out how to get a suction while you're brushing? And I said, you have a suction, but that suction is only at the dentist's office, right? It's not a portable suction that can be used. So we did a little searching and there isn't really a product outside of like a bag that you carry to pump out or vacuum out. There isn't really a solution with an electric toothbrush with automatic suction. So we made one from scratch. So we had this really interesting experience working with design engineers, working with a variety of different people to come up with a brand new electric toothbrush design from scratch. Like we designed this PCB, got it printed in China, and then shipped to us, soldered, like we made that whole thing. 3D printed and then what have you. We filed a patent on it and published a paper on this one. So yeah, it's a toothbrush that sucks out the saliva and aspirates, it collects in a little reservoir, and you dump it out. And that's one-way flow, because otherwise it'd be nasty. All right, so we're done-ish. So quick summary of what I do. We started by looking at really interesting domains that we want to target, because, you know, science and life changes very quickly, so you got to be modulatable and adaptable in your research. So whatever is interesting today, we look at a cool interaction of that, we model that, we then develop mimics or short domains that bind the same receptor. We add a self-assembling domain, because that's our jam, right? Self-assembly, localized delivery, that stuff. We then synthesize these peptides, look at binding in vitro, then we put them on cells to make sure they're cytocompatible, and they elicit the downstream signaling cascades that we're trying to elicit. We then put them in vivo for in a variety of different animal models as well as safety models. And ultimately, what we hope to try to do, and have tried unsuccessfully to do, is to translate these. We've had a couple of different startups, which I'll very, 2 seconds talk about. Yeah. So if this stuff was interesting, we have a lot of fun, cool toys in the lab. This is from the old lab, but I was very nice to everyone, and when I moved, they let me take everything. Also, I bought everything through hard-earned grant money. I made a fuss. So, we've got a couple of supercomputers, plasma synthesizer, purification equipment, a variety of different lab tools, binding kinetics tools, and in vivo surgical models as well as tools. If this stuff was interesting, if you guys are interested in the kind of research I do, please feel free to reach out. I talked about this. So science cannot be done in silos. For any junior, younger investigators, the best thing I can recommend for you to do is collaborate, right? And even for you guys, undergrads, grad students, postdocs, just find ways to collaborate to do better science. Information is free, very readily available at your fingertips, but read through it, right? Just read through it. Read through it before you send it, right? Because I have students who just AI an answer. I'm like, good, thank you, right? I could have done this too. And send it to me. I'm like, just at least read through it. Just at least read through it. Make a typo or two, you know? Anyway, so science cannot be done in silos. We work very closely with a variety of different people, both in the US, internationally, all over the place. And we think that this is a platform that needs to get translated, that we need to move forward into the clinic. And we do that through, for example, R1 that we have with Dana Graves and Shuyin Yang, where we inject hydrogels into diabetic fractures and it helps heal the bone faster, right? And that came through presentation, through going out and doing a talk just like this at Penn Dental Medicine, 10 days before they shut down for COVID. And then this toothbrush thing came through Maxine at Rutgers Dental, because I gave a talk there. Chang Leeds, another R1 that we had, I invited him to come to NJIT right when I started there, and we worked on a project to deliver his small molecules, also MGPPE, with our hydrogels into the cartilage for cartilage regeneration that he did in a rabbit successfully, and then 3D printed different structures, things like that. And then you have folks that you work with and never get funding. We keep trying to get funding, the computational work that we're doing with Peter Kraut, ND simulations, some structural work that we're doing, and things like that. Anyway, And find mentors, right? Find mentors. It's a little male-centric here, but hey, fine. Find mentors that help you out, that take you places, that you like. I took a class with this guy, but I absolutely love Andreas Garcia at Georgia Tech, right? Like, he has been an amazing mentor to me. He has written a letter for me for every single thing I've done in life, right? For my postdoc fellowship, postdoc fellowship, what is this? Faculty job, like everything. And he's been an amazing mentor. Because we have been to conference together, we've talked to each other, he's gotten to know me, right? So find mentors who you can catch up with, who can help you out, and then find people who just have like clout, right? So Rena was the head of NIDCR. She was a postdoc mentor when I was at Rice. She was at Baylor Dental, he was at Rice, and they kind of co-mentored me-ish. But then she went on to be, Fauci at NIAID, right? The dental version, and the head of NIDCR, Rena D'Souza, and just retired. Like many other people in that job. Anyway, so I'm also part of the treasurer, I think, of this special interest group. Conferences. Definitely, if you can ever go to a GRC, faculty, postdocs, graduate students, strongly recommend. People are in shorts, chatting, very casual, Nobel Prize winners to undergrads sometimes. Over beers, right? So definitely check out Global Research Conference. I love the biomaterials and tissue engineering. We're playing softball, and Andreas was behind me. He's like, "If you hit this ball, I'm not going to graduate you." It's like, "Oh no." All right, so definitely check out BMES. A lot of different career opportunities. You should go to that. I'll be at Society for Biomaterials because I'm trying to become a fellow. Right? Wink, wink. Gotta be elected. Definitely check out the Society for Biomaterials. I'm part of a special interest group there. There's a little QR code, which, the last thing, I teach a class in innovation entrepreneurship. You're not a UH, so I don't know if you care about this. All right. There is audio. So I'll tell you about the other adventures I've had very briefly. So we've had startups to look at peripheral artery disease, coronavirus, optical indications, even parking, right? So we made like an Uber for parking app, because I used to live in the Upper West Side for a little while. When I first moved to New York, New Jersey area, you just like post how long you're willing to wait and the price you want to sell your public spot for, right? We tried marketing, and then I realized it's very hard to have more than one job and be good at it. So I decided to just be bad at one job. Anyway, so yeah, thank you very much for your time. These are the people who paid for what I do. Please feel free to reach out. kumarlab.us. That was the cheapest free domain that was easy to use. That's my email address, nice and long. That's the QR code for my LinkedIn. Thank you.
Thank you, Dr. Kumar.
Thank you.
Questions?
Looking for a 5-star rating.
We have 5 minutes.
I'll start with an overview. So many undergrads in the lab.
Yeah.
Or in the,
Yeah.
we cover a lot of bases. We do a lot of things, technology development, a lot of different things. How do you choose which problems to tackle?
There are 2 main factors. I look for anything I can use the self-assembly peptide technology for that I have sufficient background, preliminary data that I can get funding for. I'm very funding driven. Or what's cool and sexy in science, right? What is on TV right now? There was talk about having specific funding initiatives for autism a couple of weeks ago by the health secretary, and I said, we do autism research, right? So we looked up a mimic, we looked up a peptide, domain, pathologic basis of disease, right? Robbins Coulter, right? And then try to work backwards from there. How can we use self-assembly or some kind of technology to treat that? Now it's very, but also we need to be realistic about what we can do, because students come to me with ideas all the time. The first thing I tell them is, how do I fund this, right? We can probably get this to, Hail Mary, great idea, no controls, one animal, but how do you fill up the gaps and how do you actually publish a paper? How do you get funding for this? But customer discovery is probably the most important. Anytime I have any idea, I first find someone's much, I don't know any of this stuff. I work with smart people, right? I find someone much smarter than me who does this a lot better than I do, and I ask them, why is my idea stupid? Yeah, no joke.
Any questions?
Every time you have an idea, like you want to go to med school, go speak to a bunch of people who want to help people, right, every day, and see what their help people is. See whether, see what pressures they have, right? I mean, who wants to go to med school? Great, right? And find out what's happening with healthcare and AI right now, right? Find out what the reality of practicing medicine. You want to be a general practitioner, you want to be a surgeon, or what have you. Like, what is the path and what is the happiness level? What does life look like, right? Don't just shadow the surgery. Shadow the day-to-day, right? Shadow the Lamborghini at 2 a.m.
So more questions? Questions? No? I have a question.
Oh, yeah, please.
So you mentioned all about like mimicking peptides, let's say agonistic peptide. How do you design antagonistic, the opposite?
Yeah, so I think the biggest challenge with designing agonistic peptides is more often than not, we would envision them to be antagonistic, right? Or binding and blocking the pocket. So we look at the activating residues of the growth factor and receptor, and we try to bind that, or we try to block that. So to antagonize, we try to block that. Now, we're not a GPCR, we're not a molecular biology lab, so we don't study all the different sites to target. So we look at existing literature of known binding sites, both antagonistic, agonist binding sites, allosteric, antagonist binding sites.
Do you use any software to design peptides?
Yeah, so a lot of software. So it's here. So to model the proteins, we use AlphaFold, RosettaFold, and then to design we design the peptides, and then we dock them onto the different binding sites using AutoDock, FlexPepDock, actually a number of other docking softwares. And folks are coming up with new versions of these different modeling softwares all the time. And this is a relatively, I don't know, couple-month-old slide, so yeah. And then we published a book chapter on this, so if you're interested, you can reach out to me. I can send that to you.
Excellent. Any more?
So like using AI machine learning, so how do you address the limitation that you're facing in experimentation?
So I think, I don't know how best, so we don't have enough data to learn to do sophisticated AI models, right? So we're trying to use artificial intelligence and machine learning to develop a new secondary structure, linear beta, how much can I say? Linear beta sheet, but there's very little data. And the way we're doing that is taking a look at specific peptide sequences that form beta sheets through libraries of existing proteins. But the problem is a lot of the time those are much larger macromolecular structures, so it's harder to learn from that to do this. Yeah.
Excellent, excellent. Give a big applause.
Oh, thank you. Oh, sorry. Right there, yeah,
Yo no tengo más cosas que and that is it. Nice to reach out to like regarding what it asks.
Okay. Okay.
I don't think
Yeah,
he's
same, yeah.
but I feel like he's has good ideas.